Good day. Lecture about acute intestinal infections, general characteristics of acute intestinal infection and their causative agent. Acute intestinal infection or acute intestinal disease are infections human disease characterized by a primary lesions of the gastrointestinal tract and intoxication. Incidence of acute intestinal infection according to the frequency and level are second only to influenza. According to WHO World Health Organization, intestinal infection cause about 1 billion of disease in the world. In many countries they comprise from 20 to 35% of the total mortality. Acute intestinal infection are example typhoid fever, paratyphoid, dysentery, salmonellosis, escherichiosis, cholera, and other infectional processes which localized in intestinal tract. We have to understand about common features of intestinal infection. First, it's this infection all-round spreading, seasonality, source of infection, entrance gates of infection, the mechanism and the ways of transmission of this infection. The pathological process is localized in intestine, are characterized by common symptoms of the disease. A variety of clinical forms of acute intestinal infection. Bacterial carrier mode. To most of acute intestinal infection, no lasting, long-standing immunity is formed. And the method of laboratory diagnosis, we used bacteriological method, isolation of pure culture and identification of microorganisms, serological method, detection of specific antibodies in serum in the process of the disease, or detection antigens of the pathogens or causative agent, and molecular genetic method we detect DNA or RNA of causative agent which cause intestinal infection. Prophylaxis, we know about non-specific and specific prevention. Non-specific sanitrogenic, armed and breaking the epidemiological chain. Identification of patient and carriers, control of water and food, keeping to sanitary hygiene standards at home and in public catering, keeping to personal hygiene. Wash your hand every day. And second, specific prophylaxis. Currently work is underway on development of oral vaccine for formation of local immunity. The following vaccines are used for prophylaxis in my country. For example, we used Vienvac, chilled walled cell vaccine, oral vaccine, live oral Ty21 typhoid vaccine, vaccine of purified V antigen Typhum V, Shigella vac is dysentery vaccine against Shigella zoonei. General properties of coliform bacteria or enterobacteria. Most causative agents of acute intestinal infection belong to the Enterobacteriaceae family. Enterobacteriaceae family is divided into groups, genera, and species. Group 1, Escherichia tribe, includes Escherichia, Edwardsiella, Citrobacter, Salmonella, and Shigella. Group 2, Klebsiella tribe, includes Klebsiella, Enterobacter, Hafnia, Serratia. Group 3, Protea tribe, includes Proteus genus. Group 4, Yersinia tribe, includes Yersinia genus. And group 5, Erwinia tribe, includes Erwinia genus. For us interesting, we study Escherichia genus, causative agent of escherichiosis, Salmonella, causative agent typhoid fever and salmonellosis, Shigella, causative agent of dysentery. What about general or common features of causative agent which cause acute intestinal infection? First, its morphology. Microorganism, small gram-negative bacilli prone to polymorphism, have common pili of the first order and sex pili second order. Due to their genetic information exchange during conjugation, we know about it. Flagella, except flagella of other type, may lose their flagella under the influence of various external factors. Many enterobacteria have a microcapsule. For your understanding, Shigella doesn't have flagella. Another microorganism, family Enterobacteriaceae, have flagella, and this organism motile. Enterobacteriaceae can't be identified by the morphological properties, because if we will prepare fixed smear, and if we use gram staining technique, we will see same pictures: gram-negative microorganism, gram-negative bacilli, Shigella, Salmonella, Escherichia. We will see gram-negative bacilli, and we can't, under microscopic technique, identify microorganism. Maybe Shigella, maybe Escherichia. We can't identify. On this slide you can see gram-negative bacilli, and we can't understand which species of Enterobacteriaceae family we see. Maybe Escherichia, maybe Shigella. Pictures will be same. Next, cultural properties. Enterobacteriaceae microorganism, species of Enterobacteriaceae family, grow on ordinary nutrient media and give the similar colony small, shiny blues. Virulence form S-form colony. Form a flat, raw, grainy form R colony after loss of capsule. Highly mobile microbes, example Proteus, manifest a phenomenon of swarming on agar. Therefore, they are cultured or on different diagnostic media. For example, Proteus grows phenomenon swarming on surface, cystine lactose, electrolyte deficiency agar. This on surface simple nutrient media you see swarming. This growth of Proteus on surface sled agar, which inhibits the Proteus swarm. For cultivation of microorganism Enterobacteriaceae family, we can use McConkey agar, and we can understand what microorganism grows on surface nutrient media. For example, on surface McConkey agar, Escherichia coli form pink or purple color of colony. Klebsiella species form pink colony. Salmonella species form colorless colony. Shigella forms colorless colony too. For example, we can use eosine misceline blue agar or entoy agar. We can use gectonic agar, and we can use Salmonella Shigella agar. On this slide we can see, for example, how grows Escherichia coli and Salmonella on surface agar, gectonic enter agar. You see Escherichia coli form yellow-orange color of colony and blue-green with black center, because produce H2S, form Escherichia coli. Salmonella. Now on this you can see different colony on surface gectonic enterogar, how grows Shigella, colorless colony, and Salmonella. We see black colony because Salmonella forms H2S. On surface McConkey agar we see lactose activity of microorganism because all microorganism family Enterobacteriaceae classified by two groups: lactose fermenting, lactose positive, and lactose non-fermenting microorganism, lactose negative. If, for example, on surface McConkey agar you will see red colony, pink colony, you have to know that microorganism lactose positive ferment lactose. And if you see, for example, colorless colony, you will understand this microorganism lactose negative. On surface eosine miscellaneous blue agar on endoagar we can see how grows Escherichia coli S-form colony with metallic green shine colony. Its characteristics of colony Escherichia coli. Shigella and Salmonella on surface XLD agar. I repeat, Shigella doesn't produce H2S, and colony will form colony colorless. Salmonella on surface XLD agar, on surface gectonic agar will give black colony because produce H2S. On differential diagnostic nutrient media for understanding between Salmonella or Shigella we used gectonic agar, XLD agar, and SS agar, Salmonella Shigella agar, and I repeat, black colony Salmonella. If you see colorless colony, it's Shigella. Biochemical properties of family Enterobacteriaceae are very pronounced and are different for different genera. Most active genus Escherichia ferments carbohydrates on GIS media. GIS media is number of carbohydrates to acid and gas. Microbes of genera Salmonella and Shigella do not ferment lactose, are lactose negative microorganism. Other carbohydrates are formed to acid or to acid and gas. The more biochemical activity, the less pathogenic microorganism. Biochemical activity of Enterobacteriaceae is used for identification. Identification begins with the first seeding of the differential diagnostic medium with lactose and glucose, and with respect to lactose, two species are distinguished: lactose positive and lactose negative. If colony on surface nutrient media which contain lactose, we see color colony, it's lactose positive microbes. If we will see colorless colony, it's which on surface nutrient media which contain lactose, it's lactose negative. Basic identification test which we used for identification of microorganism family Enterobacteriaceae: lactose, ferment of lactose, oxidase reaction, oxidase negative, fermentation of glucose to acid or acid and gas, reduction of nitrates, reaction with methyl red, Fogis-Prascauer's reaction, indole formation, urease activity, reduced citrate, H2S formation, mortality test, and another test for biochemical identification of microbes. On this slide you can see how growth of different species microorganism family Enterobacteriaceae on triple sugar ionic medium or agar clear. On surface you can see lactose negative red color and glucose positive yellow color without black color. H2S negative. On this slide you can see slant parts. It's fermentation. Place for fermentation lactose, slant lactose. In deep we will study glucose activity. Lactose negative, glucose positive and produce H2S because we see black color. On this slant parts yellow, we have to understand this lactose positive, glucose positive and produce H2S. On this slide, for example, you see the same picture, this control triple sugar ionic medium, this control medium without microorganism, this red slant lactose negative, red but glucose negative. We don't see bubble of gas, no gas, and we don't see H2S. We don't see black color. For example, yellow slant lactose positive, yellow but H2S, and we see bubble gas. Growth of Enterobacteriaceae on TCY or Kliegler's medium. You see different characteristics of biochemical characteristics. Glucose positive to acid without gas, lactose negative, and this we see hydrogen sulfide positive reaction. On this slide you can see reduction of the nitrate, how change different nutrient media, different microorganisms. For example, this test tube with control without microorganism, this microorganism Escherichia coli change slant parts from yellow to red, and another microorganism don't ferment or don't reduce on the nitrate. For example, urease activity. In this test tube we see negative reaction yellow color and positive reaction pink color urease activity. This example of biochemical test utilization of citrate. Green color negative reaction. If color test nutrient media change from green to blue, you have to understand positive reaction on citrate. Test on the mobility on Enterobacteriaceae. If you see, for example, diffuse growth of microorganism on test tube, you have to understand microorganism moving through of agar. This negative, this positive motile test. For identification of microorganisms, we can use complex entero test, which includes different biochemical tests. For example, H2S, lysines, indoles, ornithine, phenylalanine, and another test for identification of microorganisms. On this slide you can see different color, different tests of microorganisms. For example, H2S yellow white color negative. This microorganism produce H2S. Next, pathogenicity. Pathogenicity, we know that Enterobacteriaceae family, that microorganism gram-negative, and all gram-negative microorganism in cell wall includes lipopolysaccharide. Lipopolysaccharide consists 3 fragments core polysaccharide, O polysaccharide, and lipid A, endotoxin. Pathogenicity of Enterobacteriaceae family includes antigens, capsular K, flagellar H and O somatic antigen, and virulence factors, immune evasion, capsule, fimbria, type C secretion systems, factors of adhesions, factors of exotoxin formation, endotoxin 2, hemolysins, iron bonding compounds. Antigenic structure. The causative agent of acute intestinal infections have a microcapsule that contain K antigens. It has 4 variants by sensitivity to temperature: A. Thermostable antigen, B. Thermostable antigen, and L. Thermolabile antigens. Different species of Enterobacteriaceae includes V antigen capsular polysaccharide. Enterobacteriaceae contain somatic O antigen, which have characteristics like thermostable, resist by boiling for 1 to 2 hours, is most destroyed after processing with alcohol and formalin, and chemical structure lipopolysaccharide of a cell wall, antigenic determination determined, and terminal monosaccharide. Motile enterobacteria contain H antigen or flagellar. This antigen, thermolabile, destroyed by heating after phenol treatment, retain their antigen properties. By chemical structure, it is protein flagellin. Enterobacteria have common antigen for this family. Some representatives have crosswise reacting antigens, common with intestine structures. On this slide, you can see, for example, structure of gram-negative microorganism family Enterobacteriaceae. You see, for example, O antigen, which inhibits phagocytes killing, endotoxin or lipopolysaccharide produce exotoxin, for example, enterotoxin, different type of pili or fimbria like factors of adhesion, and different species of microorganisms contain V capsular antigens. Crosswise reacting antigens also belong to the factors of adhesion. They are common antigen in the representative of various species. They have similar antigen determinants, but different carriers. If such antigens are present in microorganisms, the immunocompetent cells may fail to recognize them as alien. Such mimicry phenomenon maintains the life of bacteria and the microorganism. Next, properties of causative agent in acute intestinal infection. Enterobacteria are labeled and change their properties. Example: biochemical, cultural, and antigenic. And about factors of pathogenicity or virulence factors. First group, factors of adhesions and colonization. It's pili of first order, adhesive surface proteins, lipopolysaccharide of a cell wall. They bind with the components of glycocalyx and with the membranes of epithelial cells. Adhesions is the essential condition for multiplication and increase of their amount. There are specific receptors for enterobacteria upon the mucus, a phenomenon of biological recognition. Second groups of virulence factors, factors of invasion. In gram-negative microorganisms, the factors of invasion are typically represented by the proteins of an outer membrane, by the so-called proteins invasions. Their mobility also mediates penetration of microorganisms into a sensible cells and their multiplication in the macroorganism. Salmonella mortality and salmonella mortality and multiply in macrophages of intestine, lymphoid tissue, and internal organ. Factors of invasion, it may be different ferment which produce microorganisms, example hyaluronidase, neuraminidase, fibrinolysanes, plasmacoagulase, lecitinates. Third group, virulence factors, it's factors of aggression. Factors of aggression protect of microorganism against phagocytosis. For example, microcapsule, V antigens, surface structures, ferment catalase, or crosswise reacting antigens. And last groups of virulence factors, factors of toxigenicity or toxin formation. Toxins of enterobacteriaceae, gram-negative microorganism, and we understand that microorganisms produce exotoxin and contain into the cell wall endotoxin lipopolysaccharide. Toxins are either metabolic product of microbial cells, exotoxin, or integral component of the cell wall that are released as a result of its destruction, its endotoxin, that bring in disorder into the life of the macroorganism. Characteristic of endotoxin of enterobacteria is the structure of lipopolysaccharide components of a cell wall of gram-negative microorganism. The core part of these components is realized only after the microorganism death, means destroy of cell wall of gram-negative microbes. It can produce the following effects upon microorganism. Pyrogenic effect, because endotoxin induces release of interleukins 1 from macrophage, which affects the center of thermoregulation. A toxic effect on the vessels, as endotoxin and change permeability of vascular wall. The result in a hypotonic effect, in several cases a collapsoid state. Next effect, stimulating effect on the blood coagulation systems, as it activates Hageman factors, factor 12 of the blood coagulation systems, and cause in several cases the cause syndrome of dissemination intravascular coagulation. Can cause effect of endotoxin, hepato and cardiotoxic effect, membranolabelazing effect, and effect upon the immune systems. For example, on this slide you can see what clinical features cause endotoxin and why. Endotoxin causes edema, this mechanism, this why is this effect upon microorganism. Why? Endotoxin cause different Clinical symptoms, example, fever, hypotonic effect, neutrophilic hemotaxis, and Dick effect. Dick means cytotoxin, and now we explain about exotoxin. Different species of microorganism and Enterobacteriaceae family produce different type of toxin with different mechanism of action. For example, Shigella subspecies produce Shigatoxin. Mechanism action inactivates 16S ribosome by removing adenine from rRNA. Effect upon cell of intestine tract, mucosal gangliocyte gastrointestinal mucosal cells damage, ST also enhances cytotoxin release. Shigella toxin means enhances cytokine release, causing hemolytic uremic syndrome. For example, enterohemorrhagic Escherichia coli produce Shiga-like toxin with same mechanism of action, inactivate 67 ribosome, cause inhibition protein synthesis. Enterotoxin, which includes increase fluid secretion, for example, produce enterotoxigenic Escherichia coli. Enterotoxigenic Escherichia coli produce heat-labile toxin and heat-stable toxin. By this toxin, we see that different mechanism action. Heat-labile toxin increase cycle AMP adenylate cyclase and increase secretion in gut, chlorine secretion and water in efflux. Heat stable toxin increase cycle guanylate cyclase and decrease resorption of chloride nitrate and water in gut, but effect upon intestine tract will be same: water, diarrhea. Don't forget, endotoxin of microorganism Enterobacteriaceae family cause similar effect. Different about exotoxin, we know that microorganisms produce two main toxin. Toxin of inhibits protein synthesis, and second group increase fluid secretion. Different species of microorganism produce hemolysin, is a thermolabile proteins that lyses red blood cells. Hemolysin is a pore-forming cytolysin. The hemolysin chain HLY can be localized both on the plasmid or chromosome. Hemolysin synthesis is observed in many enteropathogenic, enterotoxigenic, and uropathogenic strains of Escherichia coli. I will read lecture about different groups of Escherichia coli. Don't forget, my dear student, endotoxin produce all species Enterobacteriaceae family, gram-negative. And different species of Enterobacteriaceae family produce main two groups of exotoxin, which inhibits protein synthesis, Shiga-toxin, Shiga-like toxin, and exotoxin which increase fluid secretion, heat-labile and heat-stable toxin. Different species produce hemolysins. Thank you for your attention.